Well, you can see this is the first time either of us do this. So, but I think that's the big news. So Somesh Mohapatra, I remember reading his application letter. I've told this to everybody that will listen to me, but he wrote he was going to do an MBA and a thesis at the same time. And I loved his energy. I love his precociousness. He finished his undergrad in 3 years. So I knew he was the right hire. And then I saw the MBA things like, well, I don't think that's a thing. I don't think that's possible. That's not how the world works. Well, you know who was right and you know who was wrong. So I'm really, really excited to be another part in Somesh's precociousness, and he's going to be the first person to graduate the group this year, and we're really looking forward to seeing him step up and do the job. Thank you.
Thanks, Rafa, for the great introduction, and thank you everyone for coming to this talk. I'd be speaking today about what I have been up to in these 4-ish years, 3 years, 10 months on designing macromolecules using machine learning and simulations. I skipped the days knowingly. All right. Very well-known PowerPoint glitch. Okay, it works now. So, before I jump into the science, I want to take a step back and acknowledge a whole lot of people who made this thesis possible. But before that, let's just remove this panel up. Sorry about that. All righty. At first, my advisor, Rafael Gómez-Bombarelli, I can't thank him enough for helping me with the science, with the machine learning, with the ideas, and most importantly, supporting and letting me know when it is too crazy to do something. Also, my thesis committee, Alfred Alexander Katz and Brad Penteluti, thank you very much for being on my committee, helping me ideate as I did, and Brad for being a great collaborator, to Rafa and his students for being one of the best collaborators I could ever have. And the key collaborators here, like Carly, Nina, Joe, Sarah, I just can't name every single person in the Penteluti lab. Yasmin's here. I see a whole lot of people from whom I have learned the chemistry, the biology, the chemical biology, things that I'm not an expert in, and I won't even claim to be an expert in today. But it has been a pleasure to have interacted with you through this entire period. Thanks to the professors at MIT who helped with the data, with the understanding of the various scientific aspects of the projects that we worked on together. And also during my PhD, I interned with Google Accelerated Sciences, with my managers being Lucy Colwell and Suhani Vora. Sue is also attending this meeting. Thank you very much for helping me develop the perspective that simple models can be efficient enough and can help in making robust predictions. I owe a lot of that thinking to both Lucy and Sue. And thanks to the funding agencies, SenseTime, JClinic, Takeda, and Novo Nordisk, for whom I thankfully didn't have to TA any semester here. Alrighty. Okay, needs another click. Perfect. So the outline for today's talk. For the folks here on the side of the room, we are doing as we should. All right, we'd go through the background and challenges, then go through the approach, the methods, and the tools that were developed to tackle the challenges. We'll speak of two applications here. One from the biomacromolecule side, speaking about mini proteins and peptides for gene therapy delivery, and one from the artificial macromolecules, talking about thermosets or deconstructable thermosets. And before everything gets too much, I'll end with a quick summary. All right, so background and challenges, what and why? What is a macromolecule? An individual macromolecule is distinguished by the identity and composition of the monomers and linkages. Monomers being the building blocks of the macromolecules, and linkages connecting these individual monomers into various topologies, which can be linear, tree-like, bottle brush, anything. And where do we see them? Biomacromolecules or biological macromolecules form the basis of life as we know it, starting with strands of polynucleic acids as DNA or RNA, or even like connections of all of these amino acids. If they're short chains, then they're peptides. If they're long, then we call them as proteins. And when they start becoming very complex nonlinear structures made up of sugars or saccharide units, we call them glycans. And apart from these biomacromolecules, artificial macromolecules are just ubiquitous and indispensable to life as we know it. If you took a COVID vaccine shot, which I believe all of us did, we received it in a PEG lipid nanoparticle based carrier. And that carrier, it's itself a macromolecule or a polymer based carrier. Or if you picked up some water on your way or a coffee, there's high chance it might have been in a styrofoam cup. Or a lunch bag, a polyethylene bag. And if you love playing with Lego as much as I do, then yeah, Lego blocks are also macromolecules. So macromolecules are just everywhere, and we can't omit the possibility that we have interacted with them or just not have used them in some way or the other. So why is it even required? Isn't it a solved problem? Why do we even care about macromolecules at this point? The challenge comes from the combinatorial complexity of it. But let's do a small exercise. You have 20 words, ignore the grammar for a bit, and you have 100 positions. Let's say a 100-word long sentence. The possibilities of all sorts of sentences there, or the number of permutations, is 20 to the power 100. Each position taking in 20 different types of words. And that is of the order of 10 to the power 130. Replace the words with monomers, sentences with macromolecules. Suddenly you have 10 power 130 macromolecules. And all of these are just linear. The moment you enter into the nonlinear space, there's this wonderful formula called as Cayley's formula. You have n monomers, one bond type. You have n to the power n minus 2 macromolecules there. And if we think the same thing as 100 different monomers, we have approximately 10 to the power 200 macromolecules to choose from. So if you have one property, ideally, one out of this 10 power 100 or 200 is what you need. As a result of this chemical diversity, just finding that single macromolecules becomes really challenging. And it brings in its own sets of challenges around representing as to how do you write a macromolecule? How do you compare two macromolecules together? Or how do you build a function that can learn over macromolecules? And we would look at the challenges of each of these cases right now. Starting with representation. If you have a set of letters, okay. Yeah, this is working. Sometimes it's just finicky. Sorry. Representation. Imagine you have 4 letters and your entire alphabet set has 4 letters, A, T, C, G, like the DNA, and you have to write a linear one. You can totally do like an entire string of letters easily and be happy with that. And if you're doing something like a peptide, you can write a peptide for Thesis. Trust me, this is a synthetic amino acid and I've used all the 20 natural amino acids that I could. But there's a caveat. I'm missing the O here. Because in the set of natural amino acids, there's no monomer which is attributed to O, or there is no universally accepted monomer. So my O can be very different from what your O is. And that poses a significant challenge for making a universal representation, even for something as simple as linear macromolecules. And start thinking when you run out of the space of alphabets, capital letters, small letters, 52, we are done. After that, symbols. What happens when your vocabulary or your set of monomers is way larger than that? We are at a loss of words, literally, in this case. And when we start doing nonlinear macromolecules, it gets even more hairy. How do you represent something that has a bottle brush or a chain in it? People have tried several approaches. Professor Olson's group here does big smiles trying to do like nonlinear polymers together, having statistical things, but it's still very challenging to get a one-size-fits-all representation. There have been works, such as by Rampi Ramprasad's group, now at Georgia Tech, where they use property vectors. They said, okay, we don't really care about the individual macromolecule. We'll just have a fixed size property vector to do it, like represent the atomic level descriptors, the quantitative structure property relationship descriptors, and all of it. But the challenge is, the moment you change the type of the atoms or the monomers, you need to customize it again. So every time you change your chemical space, you're at the risk of doing a new representation. Moving on to the aspect of similarity computation, we are all very familiar with the COVID variants, with like the alpha, beta, gamma, delta, omicron, BA2 omicron, and so on. All of those variants are usually computed or like calculated using this matrix of the Blossom 62 matrix, where if you have two Cs or two cysteines, you get a +9 score. And if you have two serines, you get a +4 score with two S. You ask me why it is like so discriminative between just two letters, which are seemingly similar. It is because in the course of evolution, having a C conserved across two mutating sequences has a higher probability than having two S conserved. It has no chemical similarity as such in it. And when you move into the space of nonlinear macromolecules, there's hardly a method today that can help in computing the similarities between general macromolecules. So the methods today are very much limited to biological macromolecules. If you put in one unnatural monomer, everything breaks down, and they really do not extend to nonlinear topologies. Now going back to what a lot of us must be waiting for. So what are you doing in terms of machine learning? So we have been seeing a decent amount of reports in the literature that a new molecule has been discovered. Mark my words, it's a molecule that has been discovered, not a macromolecule. There's significant difference between those two terms. For the world of small molecules, methods such as ChemProp, based on message passing neural networks, are quite advanced, which have helped in the discovery, the property prediction and optimization of small molecules. Unfortunately, we have been limited by the representation or the descriptors in terms of macromolecules, and one of the best works is quite old in terms of like the machine learning field. It's from 2018, based on hierarchical fingerprints and polymer genome. And I say old because the field of machine learning is moving at such a breaknecking speed that 2018 seems like ages ago. So based on these three critical pieces, representation, similarity computation, and machine learning, I went ahead and developed the approaches where we will discuss methods and tools that tackled each of these parts individually. Starting with representation. A while ago, we looked at the macromolecule being made up of monomers and linkages. We use that same hierarchy to start with the representation. The monomer, or an individual macromolecule, was represented at first by the physicochemical descriptors. Starting with the molecular weight, its lipophilicity index, or if it's a molecule, how many rings are there, how many carbocycles are there, and so on and so forth. You see where I'm getting at? At a point, I will get to a place where I'll think I have exhaustively covered all descriptors, but when a new property comes in, I'd be like, oh, I wish I had that extra descriptor. So the field of these physicochemical descriptors, manually crafted, is very interesting to say the least. So we moved to a very different representation where we found a barcode or a circular fingerprint. This idea is based upon works that were done earlier in like 1965 by Morgan and like later by extended connectivity fingerprints in 2010 by Rogers and Han. So what it does, it gives the entire molecule a unique barcode or a fingerprint. And each of these positions, let's say number 132, which is the activated index, is marked for a particular substructure. And the substructure at this point is a small subgraph in this entire molecular graph. So what that subgraph is, it starts or is centered at this nitrogen atom and looks at its two nearest neighbors, the carbon here and the carboxyl oxygen, the carbon here and the two nearest neighbors up here as the two other carbon atoms. And capturing all such substructures or subgraphs, each molecule gets its unique fingerprint, which implicitly captures the chemistry, and we do not require any of these handcrafted physicochemical descriptors. Now moving from here, we start looking at a level above and look at the representation of the macromolecule itself. At the macromolecule level, if you remember, I just mentioned that there's no one size fits all. And if we have a macromolecule, which is like the chain is kept constant and there's a secondary substituent like an R1, R2, R3 that keeps on changing, we could just represent whatever is in that differentiating group and capture the entire variance of the macromolecule. Or if we have a polymer network, we could just represent the monomer information and the mole percent or the amount of these two respective things and be able to say that we have represented the macromolecule accurately or like decently enough. However, when we move into the space of peptides, which are sequence-defined macromolecules, at that time, we used a fingerprint matrix representation. In this case, we got all of these fingerprints stacked as they would be on the backbone of a peptide or a protein. And extending this idea to the nonlinear space, when we came across these macromolecule graphs, we represented the monomer nodes as the fingerprint and the linkages, which are the functional groups of the bonds that connect these monomers using the fingerprints of these functional groups. So essentially developing a representation that was able to capture any general macromolecule. And now touching back, how do we handle the similarity computation? We used the same approach of two macromolecules being aligned together. But at the same time, instead of using an evolutionary statistics matrix, we developed a Penimoto similarity matrix or a chemical dissimilarity matrix, where we compared as to how similar the fingerprints of individual monomers were. So the scoring was changed to represent the chemical similarity instead of evolutionary statistics. And that helped us to incorporate any unnatural monomer we might have in the future, any chemistry that we might require, and just extending the same idea again to the nonlinear space, we were able to do so when we wanted to compare a linear macromolecule to something that looked like a ring, or a linear to a branched, a linear to like a bottle brush looking shape. And this entire thought process was an advancement in the field which helped us to move into the space of developing a generalized similarity computation method for two arbitrary macromolecules of known monomer composition and topology. Now going away from the similarity computation to the part of machine learning. Again, the machine learning was not something like a one size fits all. Here we put three metrics into place, starting with the data type, the data set size, and the task. If we had something like just linear macromolecules and a very small data set with the differentiating groups, then we could go for a physics-informed model. or if we had data which were like less than 100 data points, we could use the simpler models. However, if we wanted to go into the mid hundreds, like hundreds to thousands, 500, 600s, or even greater than that, where the data set size was enough to do something like a CNN or a graph neural network over nonlinear macromolecules, we moved to those model architectures. And depending on the task, all of these models, the physics-informed, the simpler models, CNNs and graph neural networks, GNNs, were based on doing classification and regression. But if we wanted to discover new macromolecules or molecules in general or sample new molecules, we used a language-based model or a recurrent neural network. So with this, I end this subsection on the approach, methods, and tools and move on to applications. I have been really lucky to apply these tools to systems ranging from peptides and glycans to electrolytes and thermosets. This is a summary slide of all that I have applied these tools to. In bold are the papers that have been published. The gray colored notations are for the key conferences that were a part of these publications. And some of these are like manuscripts that are under preparation or the experimental validation is undergoing. As a quick overview, a whole lot of the work, like the entire top row, was in collaboration with Brad's lab, starting with peptides and mini proteins for gene therapy delivery, one of the things that I'd be talking about today, finding peptidomimetic binders for cancer and other target proteins, then discovering cancer vaccines, again, peptide-based cancer vaccines, optimization and prediction of automated flow synthesis for peptides and peptide nucleic acids, taking these tools to nonlinear macromolecules with prediction and optimization of glycans and antimicrobial peptides, some of it with Professor Laura Kissling's lab, then doing oligomer electrolytes with Yang Xiaohan's and Jeremiah Johnson's labs, then deconstructable thermosets with Professor Johnson's lab again, and inverse design of molecules, where we extended our toolkit to take care of small molecules, where we did something with organic photoelectronic molecules, and did a pretty interesting project where we tried to do inverse design from shape to molecules. So given a random shape with desired chemistry, we could generate a molecule directly from there. So now I'll be moving into the subsection of talking about the biomacromolecules of gene therapy delivery and deconstructable thermosets. This work was done in collaboration with a whole lot of people from the Pensaluti lab. Carly, who is on the West Coast now, Eva, Andre, Rafa, and Brad being the key players in this entire project, working on prediction and optimization of gene therapy delivery. In each of these subsections, I'd be moving through a format where I would talk about the motivation, the data set, the machine learning and the experimental validation, the interpretation of the machine learning model. So these four these four steps would be a part of each of these subsections here. So starting with the motivation of this. The motivation for this gene therapy delivery started by looking at diseases or rare genetic disorders such as Duchenne muscular dystrophy, which leads to an absence of a key protein in human muscle tissue called dystrophin. So instead of the healthy muscle tissue looking like this, the Duchenne muscle tissue has fats and fibroids in them. In mid-2010s, around 2015, Sarepta Therapeutics developed this medicine or a therapeutic called eteplirsen. Eteplirsen, interestingly, was supposed to be given at a significantly high dose to the human body. Like if you think just the numbers as 1 milligrams per kilogram of Advil, a kilogram of body weight, what you'd be taking, eteplirsen was being given at like 1 gram or 10 grams per kilogram of body weight. So about like 2 to 3 orders of magnitude easily. And that was a huge challenge when it was being fed or like injected into a kid who was 3 years old or something. So they had like significant nausea, vomiting, and all other side effects which were undesirable. So an approach to improve upon that was to deliver this oligonucleotide or an antisense oligonucleotide, which was the key therapeutic, using mini proteins or peptides. So by conjugating a cell-penetrating peptide or mini protein to it, which would help in increasing the delivery of this oligonucleotide into the cells, it was hoped that the delivery could be improved and thus the dosage could be decreased accordingly. But the challenge remains again. What is the most optimal peptide sequence that you can think of? It's the same question we go back to. 100 positions, 20 amino acids, 10 to the power 130 possibilities, and you can get one. So we looked at the data set. Thankfully, very painstakingly, an entire data set of 600 modular mini proteins was put together by students at Brad's lab, Justin Ulf and Colin Fadson, where they had mapped each of these 600 modular mini proteins to their phenotypic readouts by testing them individually and had a number referred to as mean fluorescence intensity for each of these individual sequences. So 600 sequences, each sequence had a number associated to it on a scale of 1 to, let's say, 20, which was the maximum of the range of the training data set, which said that how better each of these sequences performed as compared to the drug alone. So if it is 20, it's a 20-fold improvement in terms of delivery. On this data set, we used a generator predictor optimizer framework to design optimal mini proteins. The generator was trained and was used to generate new peptides. The predictor was trained on a fingerprint matrix representation on the activity. So it mapped this fingerprint matrix to the activity, and seeding sequences obtained from the generator to the predictor optimizer loop. We increase the activity, decrease the similarity, essentially trying to get novel peptides in the process, and decrease arginine content, because arginine is known to have toxicity effects when it is injected into the mice models and subsequently into humans. By using this framework, we discovered 13 unique peptides. And these 13 peptides, or the MAC peptides, were carried forward to be tested experimentally in vitro in cells. And subsequently, some of these were selected to be tested in vivo as well. So here are the results of the model. On the test dataset, or the held out test dataset in the machine learning model framework, which were essentially not shown to the model when the model was trained, we had reasonable predictions on that, about an RMSE of 0.41 or a root mean square error of 0.41 of the standard deviation of the training data. And the MAC peptides, the model was decent enough, which was able to go well beyond its maximum of the training data and predict sequences which had activity as high as 50 in some cases. And what was most critical in this part was the framework was able to discover peptides that were most active amongst all cell penetrating peptides known at that point. Like the library had a maximum of around 20, and some of the peptides that we saw in the literature were around 30. And we got many proteins that were 50-fold active in helping deliver this oligonucleotide therapeutic. And at this point, we were pretty curious as to what is the model seeing in these peptides that make it make such decisions. And that curiosity led us to look at this from an attribution analysis perspective, or interpret the decision-making process of the model. So looking at this input matrix that we had, or the feature matrix, we looked at all those features that triggered the model to make such a prediction. And by averaging across these fingerprint indices, we looked at the residues or the motifs, which was more critical to the model. Anything that has a high activation is essentially contributing more to the MFI or the activity for that particular peptide. So we looked at one of these particular amino acids or residues, X, which is an unnatural residue, aminohexanoic acid, and just looking at its entire structure, we could go back and find what substructure matters more to it. And the substructure was the alkyl backbone in this case. Such level of interpretation and machine learning, where we could go back and look at the exact residues and the substructure that led to this prediction and the decision making, was not possible before this work. Now, a lot of these peptides or many proteins that we looked at were way more than 30 amino acids. And for challenges based on the chemistry and the biology, we really wanted to decrease this length to something like which was more palatable, like 10 amino acids, 20 amino acids. But looking at our data set, we had, looking at the data set, it was like almost impossible to do so, because our data set had a mean length of around 42 amino acids, and our desired design space was less than 20 amino acids, which did not have like peptides which were like as active as we would want them to be. So we used a trick. We used a trick referred to as data augmentation. Instead of this blue curve, which the data set was, we showed a bimodal distribution to the machine learning model. We showed it multiple times of the peptides that were shorter in length, such that the model was biased to knowing like what shorter peptides look like. And at the same time, we showed it an equal amount of times of the longer peptides, which were more active in general. So the model was biased towards designing shorter but efficient peptides using this data augmentation approach. And in this way, we were able to discover a critical peptide of about 11 amino acids with a 22-fold or 25-fold activity actually. Now, to summarize this entire portion, we started with an experimental library of 600 mini proteins, trained a model, a CNN-based model with a fingerprint matrix that led to the activity prediction. We achieved reasonable accuracy, like predicting on the training, on the test data set, and also on the MAC data points that we designed using the generative predictor optimizer framework. We interpreted the decision-making process of the model and also repurposed the model to design shorter and highly efficient CPPs. Now that's an end to the first portion on biomacromolecules. Now we would move into the portion on artificial macromolecules based on deconstructable thermosets. This work was done in collaboration with the Johnson Lab. I see Yasmin here, thank you for attending. And with Peyton, Keith, Rafa, and Jeremiah as the key players in this. Now we'd follow the same structure here as well. The motivation, the data set, the machine learning, and the validation. Starting with the motivation. This curve is something that actually pushed me into thinking about this problem more. And this curve is about the amount of plastic that has been produced ever since it started going commercial early in 1950s. More than 50% of the plastics produced has been since 2000. And as you can see in this pink region, we are growing at a breakneck speed. Our usage of plastics and the production is not going to slow down in the coming future. And sadly, a lot of these plastics are non-recyclable, even today. And they end up in landfills, just increasing the waste production year on year. Most of these plastics that we would use are usually the thermoplastics or thermosets. And these thermosets are hard plastics. I'd give a quick primer on thermosets because that is what I'm going to talk about. So getting into thermosets, they start as individual polymer chains, which are made up of these co-monomers. There are some multifunctional co-monomers, which can also form like another arm other than being present in the single chain. So they can start another arm and bring these individual chains together, like increasing the density of what the macromolecule set is entirely, and also being more rigid as an end product. Now, this cross-linked chains or the thermoset has excellent properties at that point. Like they can be used for high temperature applications, looking at 180C, 200C. They can be used decently there, have good mechanical properties. But recyclability takes a hit. It's really challenging to either reuse these thermosets or even recycle them. Interestingly, a couple of key innovations came into the place in like 2020 and 2021. Starting with Johnson's lab, introducing something known as cleavable comonomers. So cleavable comonomers were introduced as another set of comonomers into these chains, which ideally would not have affected the high temperature too much, but help in recyclability. And they went a step ahead and developed another class of thing called as a cross-linking comonomer, which would help in cross-linking, but could cleave them and improve recyclability further. What started as a very good-natured and an innovative approach of increasing recyclability, unfortunately took a hit when it came to glass transition temperature. It decreased the maximum temperature at which these polymer end products could be used. So the challenge was tackled in another framework, which started with the experimental synthesis, with the base as the dicyclopentadiene or DCPD, including this cleavable comonomer in a standardized experimental process, and getting this entire data set, which was then handled through some pretty interesting and innovative machine learning approaches. And we use this model to get do virtual screening of over 300,000 possible cleavable comonomers to get 2 compounds from 300,000 and then going back to test them in experiments again. And this approach starts with the data set, as always. In the data set, we started with the first parts as the cleavable comonomer, which are ethylene norbornene and isopropyl silyl ether, as you can see them here. And then we had some degradable and non-degradable cross-linker. We had an entire suite of them. At first, we had HDMN or hexahydro dimethanonaphthalene, and a norbornene separated by a short chain ester in this case. As you must have figured out, I came here because I really don't remember the names of these molecular compounds. Getting HDMN to remember, I did try that, but didn't happen. Then we had some synthesis conditions as the initiator. The Grubbs first generation or the Grubbs second generation initiator, depending on which one was used for that particular synthesis, and some monomer initiator ratio of how well the entire synthesis could go on. After again a painstaking experimental synthesis and data acquisition over a whole year or maybe even more, we got to 101 data points. And 101 data points falls in the low data regime. Doing machine learning over just 100 data points, we had to be creative and innovative on how do we do that. So we went back to the similar representations because those were really nice, with the comonomer and crosslinker as fingerprints or descriptors and Grubbs initiator as one hot encoding. If it's 1, so it's a 1, 0. If it's Grubbs initiator second, so it's a 0, 1. And a monomer ratio as a continuous number. All of these vectors were put into a set of machine learning models, which included some underfitting linear models and some overfitting multilayer perceptrons or feed-forward networks. A combination of models which were both underfitting and overfitting in a method where we had hyperparameter ensembles and a model architecture ensembles. We got about 50 distinct models which were able to have a decent prediction of the glass transition temperature. And how that looks at this point, we were able to get the unitless RMSE to 0.3 of the standard deviation of the training data set. Otherwise, 15 degrees Celsius. And 15 Celsius, the RMSE is slightly biased because it is looking at these data points which are far off from where the maximum amount of the training data set is. So looking at the Pearson's correlation, we are at 0.96 when we compared the experimental TG and the predicted TG. Then again, we tried to go back and look at, so what is the model even thinking? And we had just like 100 data points. So we wanted to really know what the model was thinking. What is the model looking at? The model obviously was looking at the co-monomer and the cross-linker mole percentages. If the co-monomer is low and the cross-linker is high, the density is lower. It's a more rigid thing. So the TG is higher, which makes sense. But the model also said that it was looking at the partial charges and the electrotopological state index. Some help from Yasmin came in there, and there was understanding that maybe there's a Zwitterianic system that is being formed. There's a positive set of charges and the negative set of charges which are coming close and decreasing the free volume, and hence decreasing the density and increasing the TG. And when it does that for an aryl substituent in a co-monomer, then there's pi-pi stacking and decrease in the free volume there. So the model was doing something in its own basis and finding features that help it make the decision of predicting TG reliably. On the basis of that, we really wanted to make sure that the model had understood the entire composition landscape. So we did a grid search over 0 to 30% of the co-monomer isopropyl silyl ether and another cross-linker DDMS from 0 to 30%, and looked at what does the model see in terms of trends across this entire set. If you increase the cross-linker to 30%, it should have the highest TG, which was slightly greater than 180. And if you increase the co-monomer to 30% and no cross-linker, it should get to the lowest TG in this case. So the model really understood of what this entire co-monomer cross-linker composition landscape's glass transition temperature was, which was very satisfying. And taking data points from this landscape, we went back to the lab, and Yasmin validated them in these experiments, and it turned out that yes, the model was able to actually predict them reasonably well. And this was the point when we were like, okay, can we discover new molecules? Can we discover new things that can help in making more deconstructible thermosets? So doing virtual screening over, as I mentioned, well over 300,000 data points or like molecules, we found just two, PHSI7 and LinF7, which are pretty high up here with like an experimental TG reaching 179, 180 Celsius. So these are deconstructible or recyclable and really do not like affect the property that we care about. So these thermosets now can be used instead of non-recyclable thermosets in high temperature applications. So that's a key thing that we discovered from this process. And while we were doing this work, the Johnson lab had started working on a class of cross-linking co-monomers, and we retrospectively validated a whole bunch of these molecules which the model had never seen onto the data set. And we found that the model is like able to capture well enough for some of these cases, except one. And looking back on NBMESI and seeing like, why is the model not able to do so? We found out that the model had always had some amount of polydicyclopentadiene in it. There was never a condition when a co-monomer was 100% or a cross-linker was 100%. While in this particular case, the model was seeing for the first time a cross-linking co-monomer that was 100%. The model was able to give a prediction, but at the same time say it is too uncertain about it. And that is the key advantage of using a model like this. It provides the mean value, but it says that, oh, this is pretty new, and I'm really uncertain about how it looks. So the huge error bar, as you can see here. And at this point, we end this part. Oh, yeah. On the primary system, starting with PDCPD, a co-monomer, a cross-linker, Grubbs initiator, monomer initiator to predict glass transition temperature with 101 data points and a model ensemble, doing a TG prediction for a mole percentage sweep across co-monomers and cross-linkers using virtual screening to find two monomers, and then finally experimentally validating all, identifying key features that matter. A quick recap into the applications. this is all that I worked on. I have summary slides for each of these. So if you feel strongly about knowing about one of these things or more, I'm happy to go through it. And as I promised, it's a one-slide summary. In this entire thesis, I worked on chemistry-informed representation of monomer and macromolecule. Did similarity computation for two general macromolecules with arbitrary monomer composition and topology. Developed machine learning methods with attribution or interpretability and optimization or screening for various applications from peptides to thermosets. The hope is that a whole lot of these works which have been in the space of pharmaceuticals may one day help patients. It's not there yet. I really hope some of these get translated sometime in the future. But at the present, the hope is that it influences, inspires, motivates, or it gets translated into labs for like chemists and biologists, chemical biologists, and moves into the space. In the end, a thank you to RGB lab members, Gómez-Bombarelli lab members. Thank you for being one of the best support systems. Thanks to the LGO team. This was in Tucson, Arizona, standing on a Caterpillar equipment, one of the most fun memories. My parents, my younger brother who is here. Baltic Petrols, some of them here. This is the Friday night crew. Thanks for keeping me sane all throughout the PhD. And friends from back home and many, many more people for helping me complete, or like almost complete this PhD. And that's it for designing macromolecules.